Hello, good afternoon, everyone. Thank you so much for joining today's program inside our biobank, FPN's Peripheral Neuropathy Research Registry, presented by us at the Foundation for Peripheral Neuropathy. I'm incredibly excited to welcome our expert speakers that we have today, Dr. Ahmet Höke and Adam Halper, to talk to us about how our Peripheral Neuropathy Research Registry, the PNRR, as we sometimes refer to it, biobank, is helping move neuropathy research forward. They will share what a biobank is, why the PNRR was created, and how patients play a key role in its success. You'll also hear about some exciting discoveries made directly from the biobank, ongoing research projects, and how this important work is bringing hope for better treatments and peripheral neuropathy. Next slide. I'm Amanda Homscheid, Program Manager at the Foundation for Peripheral Neuropathy, and on behalf of our team, our board members, and our volunteers, thank you again for joining us and for trusting us to be part of your journey as you navigate living with peripheral neuropathy. Before we begin today's program with our excellent speakers, I just want to quickly remind everyone of a few logistical items. The first thing being that yes, we are recording today's live program. It will be emailed to all of you who have registered within 24 hours following the end of the program. It will also be permanently housed on our website, along with the presenter's slide deck for future reference. Next, second, for those who are in attendance live today, you all, will have some time at the end of the presentation for a short live Q&A. In order to submit your questions, please do not use the hand raise feature during the presentation for the live Q&A. Instead, please use the questions box in the dashboard of this platform to ask any questions. However, please focus on asking questions that are general in topic and nature. We will not be able to answer any personalized medical questions. Rather, we'll answer questions that are helpful for the greater audience. We'll do our best to answer as many questions as we can. Lastly, we do offer closed captioning for those who are in need, and that can be switched on in your dashboard of GoToWebinar. And now I'm pleased to welcome and introduce our experts today. First, we have Dr. Ahmet Höke. He is a neurologist from Johns Hopkins and is the lead investigator of the PNRR. At Johns Hopkins, he's a professor of neurology and neuroscience, director of the neuromuscular division, and director of the American Peripheral Neuropathy and Nerve Regeneration Center. Dr. Höke's clinical practice and his clinical research focus on idiopathic, toxic, and inflammatory neuropathies. He's the principal investigator of several natural history studies aimed at developing biomarkers and understanding genetic underpinnings of peripheral neuropathies. He is also the recipient of several awards, including the Derek Denny Brown Young Neurological Scholar Award and Wolf Neuropathy Research Prize from the American Neurological Association. He is also an ex officio member of the board of directors of the ANA and is on the board of directors of the PNS. Next, we have Adam Halper, who, in addition to being an FPN board member and a neuropathy patient and PNRR patient, is the head of product innovation at Elevance Health, a Fortune 20 healthcare company. At Elevance, he leads a portfolio of work with leading healthcare entrepreneurs, including running the company's Entrepreneur in Residence program, commercializing partnerships with high-growth startups, and analyzing new investment opportunities. Prior to joining Elevance, he served on the social innovation and sustainable finance team at the Green and Healthy Homes Initiative, where he developed innovative financing models to fund programs addressing the social detriments of health, with a primary focus on asthma and aging in place. Before this, Adam served as a strategy consultant at McKinsey & Company, advising clients in the healthcare and high-tech industries. Prior to McKinsey, he earned a JD from the University of Michigan Law School and an MBA from the University of Oxford, where he also served as the portfolio manager for the Business School Venture Fund. Again, we are extremely lucky to have both Adam and Dr. Höke here to speak with us. And with that, I'm excited to pass the virtual mic over to you, Dr. Höke. Thank you.
Thank you, Amanda, for that very nice introduction. Let me make sure I can advance the slides. Good. So today what I thought I would do for our audience is give a very brief introduction to what peripheral neuropathy is and then talk more about the registry and how it's improving the research that we're doing with peripheral neuropathy patients. As many of you know, peripheral neuropathy is a very generic term. It just means disease of the peripheral nerves. And depending on what type of peripheral nerve fibers patients experience, they have different types of symptoms. And the peripheral neuropathies can affect either the axon, which is like the electrical wiring in your peripheral nerves, the main extension of the nerve cell, or the insulation around it, which we call the myelin. And depending on what type of neuropathy that you have, you may have a more involvement of the axon or myelin, but majority of peripheral neuropathies, I would say more than 99%, primarily affect the axon and causes axon degeneration that is currently irreversible. So I think there's a bit of a delay in the advancing the slides, so I apologize for that. Basically, as I mentioned, really peripheral neuropathy is a dysfunction of the peripheral nerves. One of the limitations of why we do not have any disease-modifying therapies in peripheral neuropathies is that we really have limited understanding and translation of preclinical work to clinical stage because we have limited understanding of why the nerves degenerate in different diseases such as diabetes or chemotherapy, and that we have even more limited understanding of how the peripheral nerves can repair themselves and regenerate. And currently, apart from very rare type of autoimmune neuropathies, we do not have any disease-modifying therapies. And this is also compounded by the fact that the funding for peripheral neuropathy through the standard institutional funding, such as National Institute of Health, is dismal compared to other neurodegenerative diseases such as Alzheimer's or Parkinson's disease. And this is especially important considering that peripheral neuropathy affects almost up to 30 million patients in the US. This is more than all the other kind of big name neurological diseases such as Alzheimer's and Parkinson's combined. And yet we're spending less than $3 per year per person with neuropathy on neuropathy research. And that's one of the reasons why I think we do not have a big group of researchers who are doing neuropathy research, and we do not have treatments for it. And I think foundations like Foundation for Peripheral Neuropathy and research initiatives like the PNRR is really trying to change that. As I mentioned, because of the low funding, we do not have adequate number of young investigators. And when there's not enough research coming from the academic centers, there's a lack of pharmaceutical company interest and limited drug development. And I'd like this cartoon where it says, unfortunately, there's no cure. There's not even a race for a cure. And this is the way I feel in peripheral neuropathy, but hopefully we can change this. So with that, I want to introduce what PNRR, the Peripheral Neuropathy Research Registry, is. We started this more than 10 years ago when the foundation was looking for guidance on how we can improve the research, especially in the two most common forms of peripheral neuropathies, diabetic peripheral neuropathy and idiopathic peripheral neuropathy. There was ongoing research in diabetic neuropathies, especially with interest from the diabetes community, but literally nobody was researching idiopathic peripheral neuropathy when we first started the Peripheral Neuropathy Research Registry. And so we decided to kind of mirror what had been done in some of the other neurodegenerative diseases, such as Alzheimer's and Parkinson's disease groups, where we could start better characterizing the patient populations and collecting blood samples for future genetic and blood biomarker studies. And with that, initially it was 5 centers that started this, and now we have 8 centers enrolling patients for this effort. And so what is PNR? Basically, we are enrolling patients that we see in our clinics that are referred to us for their regular clinical care. Majority of these patients have idiopathic peripheral neuropathy. The second biggest group is diabetic peripheral neuropathy. And early on, we also had HIV-associated peripheral neuropathy and chemotherapy-induced peripheral neuropathy patients. Although the foundation a couple of years ago, together with the investigators, made a strategic decision to focus more on idiopathic and diabetic peripheral neuropathy, given that the incidence of HIV neuropathy has declined tremendously in the last decade. And there are other efforts for the chemotherapy-induced neuropathy. So we thought we could put our focus more on the big unmet need, which is the idiopathic peripheral neuropathy population. As you can see, majority of the patients that we enrolled so far have been in the idiopathic patient population. So what do we do with these patients? After we see them, we do a very careful history and collect a questionnaire about their symptoms, their associated illnesses, family histories, and so forth. And we also do a standardized exam that has been standardized across all the sites. And this is very important when you're studying a disease as varied as peripheral neuropathy. If your data isn't standardized and if you don't collect the same type of data, the results from any future research is going to be uninterpretable. And so we really worked hard on making sure that our data collection is as standard as possible. Many of these patients also undergo nerve conduction studies, and if clinically indicated, they have a skin biopsy, but also we collect information about their laboratory testing and if they had any genetic testing in the past. And then a big component of this effort is not just collecting clinical information, but also collecting 2 extra tubes of blood for future genetic studies, analyzing their DNA, as well as using it for biomarker studies to understand questions such as why some people with peripheral neuropathy develop pain and whereas others don't develop pain. So far, research from the Peripheral Neuropathy Consortium resulted in 10 publications, many more posters and abstract presentations at scientific meetings. And as you will see from the publication years, we're in an upswing in terms of the research output from this effort. The first probably 5-7 years were really focused on collecting enough number of participants so that we could have enough statistical power in any of our analysis. So I'll start with some of the published literature and the research that was done. In fact, one of the first papers is now 8 years ago at the early stages of the registry, where we had only about 500 samples in collaboration with one of the pharmaceutical companies, Bristol Myers Squibb, which was studying the importance of this one channel. It's called voltage-gated sodium channels that are present in our pain-sensing nerve fibers, and whether mutations or variants in these genes that encode these sodium channels could be contributing to peripheral neuropathy, especially painful type of peripheral neuropathies. And basically, this study told us that, at least in the registry that we had collected, although there was some patients with variants of what we call unknown significance in these sodium channels, the distribution of this mutations was similar in patients with pain and without pain, suggesting that these mutations was not really responsible for the pain sensation. The next paper was when we had about 1,000 patients collected by that time. It was basically describing the cohort and this registry so that other investigators at other institutions could be aware of this and that start accessing the data set. So this was more of a descriptive paper explaining what type of data we were collecting from the patients and what type of blood samples were available. This was followed by a couple studies trying to understand the importance of some of the things that we tell our patients. For example, we tell patients exercise is good and refer to some of the clinical trials done in diabetic patients with peripheral neuropathy. And this basically told us that in this paper, patients who were exercising had actually reduced pain compared to the ones who weren't exercising. And this was done in the idiopathic peripheral neuropathy patients, suggesting that the data that we knew from the published literature in diabetic neuropathy patients also applied to the idiopathic peripheral neuropathy patients. And I've been whenever I see a patient with idiopathic peripheral neuropathy, I always counsel them about the importance of exercise in reducing their pain and discomfort. Another question that we were able to answer with this study was in the literature, we knew that vitamin B6 is an important vitamin for peripheral nerve health. So if you have very low levels of vitamin B6, it can cause neuropathy. But also we knew that too much vitamin B6 can also be toxic to the peripheral nerves. And in fact, over the years, I've seen a couple of patients who were taking mega vitamins and who had very, very elevated levels of B6 in their blood, and their neuropathy was because of that, because when they stopped taking B6, their neuropathy improved. But the question was always in the literature for people who are taking, for example, multivitamins or taking only B complex vitamins, if their B6 levels are elevated twofold or threefold above normal, did that cause neuropathy or influence the severity of neuropathy? And basically, the paper showed that it really did not matter. So unless you're taking 10 tablets of mega B6 vitamins, you're not going to get high enough concentrations of B6 in your blood to cause neuropathy or worsen your peripheral neuropathy. The next study was, as I mentioned, one of our original goals was trying to better understand why some people with peripheral neuropathy develop severe pain and others do not develop much pain. They just have numbness. And so we use some of the more modern techniques, looking at lipid profile in their plasma and their blood samples, looking at their proteins that cells secrete, as well as what we call the metabolites, small molecules that are present in your blood, and looked at patients who had really severe pain. So these patients rated their pain on a scale of 0 to 10, usually 8, 9, and 10, versus the ones who had no pain. And the idea was really to see if there is a signal, even with this 60 patients. And then after we found some important differences, we validated this in additional 60 samples. And so basically, we've now have found that there's a group of plasma proteins, especially involved in what we call the complement pathway, seem to be increased in patients with painful neuropathy versus the painless idiopathic peripheral neuropathy. So this is interesting, and it may actually be quite relevant because complement activation seems to play a role in immune-mediated neuropathies, and multiple companies are also developing complement inhibitors to treat those immune-mediated neuropathies, and whether they can also have a role in treating the painful component of peripheral neuropathy needs to be investigated. And then another important question in terms of general health and vitamin supplements in patients with peripheral neuropathy. Some of you may have seen newspaper articles talking about the importance of vitamin D in general health, especially in bone health, but also vitamin D levels are important for patients with, for example, multiple sclerosis that affects the brain and spinal cord. And so the question was, and this was unknown in the field, does it also affect patients with peripheral neuropathy? And basically, we looked at patients who had vitamin D levels that were below the normal range, and we did not see that their neuropathy was any worse than the ones who had normal vitamin D levels. So vitamin D deficiency by itself is not sufficient to cause neuropathy or make the peripheral neuropathy worse, suggesting that unlike some of the other diseases, vitamin D does not seem to play a big role in peripheral neuropathy. Another question is, we know diabetes is a big contributor to peripheral neuropathy. But there are also patients who have what we call metabolic syndrome. These are people who might be obese, who might have prediabetes, or who may have elevated lipids in their blood, for example, elevated cholesterol levels and so forth. And the question was, when patients have these kind of other systemic illnesses, does that affect their peripheral neuropathy? And the bottom line was that yes. So if you have peripheral neuropathy, and if you have diabetes, or if you had metabolic syndrome, you tend to have more severe neuropathy, as well as higher prevalence of pain. So this one of the other goals of our registry is really trying to understand the causes of idiopathic peripheral neuropathy. You know, when you go to see your doctor, they do all the tests, including perhaps some current genetic testing, and you know, they run everything that they can think of. And yet at the end, they tell you that you have idiopathic peripheral neuropathy, and that's about 30% of all of our patients. And so what does it mean to have idiopathic neuropathy? Obviously, there's some cause for it. We just don't know it. And so together with colleagues at Washington University, we worked on developing a computational algorithms to better analyze the genetic data that we can do by doing what we call whole genome sequencing. So this initial paper was basically focused on, it was a kind of a white paper describing the methodology, and then we analyzed about 900 of our first batch of peripheral neuropathy research registry patients. And this is currently under review at one of the journals, but we've also deposited into medical archives as a preprint. And we found that these patients have a mutation in a gene called RFC1 that is not typically picked up with standard genetic testing. And this was an important revelation because these patients, and again, the incidence is still small, so it's about 3% of these patients have what we call biallelic mutation. That is, both copies of their genes of the RFC1 are abnormal. So out of all of our 30%, 3% of those patients have mutations in this. But the other thing we found was that even if you had only one abnormality, one abnormal gene of this, you had a higher incidence of peripheral neuropathy compared to the general population, which has this only about 2.5%. So it's possible that this could be a relatively common cause of peripheral neuropathy or idiopathic peripheral neuropathy. And so we're actually expanding this study. Unfortunately, doing whole genome sequencing for each participant is quite expensive. It costs a couple $1,000s per sample. So you can imagine if you just multiply this with the 800 patients we've done. But we're hoping to complete this study and sequence all of our participants in the PNR. Another important research is focusing on kind of what we call a biomarker in our field. And what I mean by biomarker is that, you know, when I can see a patient with peripheral neuropathy, I can examine them. I can also do a nerve conduction study or do a skin biopsy and kind of get an objective measure of their nerve damage. But the best way of determining if somebody has how much damage they have is actually looking at blood levels of this molecule called neurofilament light chain. And what this is a protein that is present in all of our nerve cells. So if you have a degeneration, this protein leaks out of your nervous system and into the blood, and we can measure it. And so this has become very important in certain diseases, neurodegenerative diseases such as Alzheimer's, Lou Gehrig's disease, but we're also finding that it is also elevated in peripheral neuropathies. And why is this important? Because as we move into developing therapies and you have clinical trials, you want to be able to measure an objective way of determining, is your treatment preventing the axon degeneration? Are you promoting nerve regeneration? So you need these type of blood markers or biomarkers to be able to more confidently determine if your treatment is working or not. So those are published papers, but we have many more projects that are ongoing, and I'll highlight a couple of these. One of them is in collaboration with Dr. Brian Callaghan from University of Michigan. We're looking at are there differences in the way the subtypes of peripheral neuropathy in patients with the RFC1 expansion. Another study is we're delving deeper into the neuropathic pain, looking at diabetic patients. So our published study was in idiopathic peripheral neuropathy patients, but Dr. Daniela Manicella from Northwestern University looked at the diabetic patients with or without pain. And in her research, she did not find any difference. And unfortunately, because this was a negative study, she's not planning on publishing it. Another researcher from London, Dr. Andrea Cortese, he is doing whole genome sequencing on a very subpopulation of patients with chemotherapy-induced peripheral neuropathy. These are patients with mostly colon cancer who received a neurotoxic drug called oxaliplatin, and he is trying to identify if they have some sort of a genetic predisposition that made their neuropathy worse. Another research on chemotherapy-induced peripheral neuropathy is the work from Dr. Stephanie Geisler at Washington University, where she is comparing the chemotherapy-induced peripheral neuropathy and diabetic peripheral neuropathy. Unfortunately, partly because the chemotherapy-induced peripheral neuropathy enrollment was halted, we may not have enough number of patients to really move this research forward. Another big study is together with colleagues and University of Cambridge, Oxford, and London. We're doing another group of whole genome sequencing, mainly focusing on diabetic and chemotherapy-induced peripheral neuropathy, mainly trying to, again, understand, are there genetic risk factors for why some people with diabetes, for example, develop neuropathy and yet others don't develop neuropathy. And those type of genetic studies can help us better understand the risk factors for neuropathy, but also potentially offer some new therapeutic targets. I just want to highlight this one study. This is not published yet, but we're working on writing the paper. And so basically, the left side are different components of the metabolic syndrome. And these are the types of symptoms and examination and nerve conduction study findings that we found in these patients. And the biggest thing to take home is that of all the metabolic risk factors, the most important thing that really determines the severity of the neuropathy is really elevated lipids. So for those patients with abnormal lipids, it's very important that if you have peripheral neuropathy, you want to get that under control. So uncontrolled high levels of triglycerides is a huge risk factor for worsening peripheral neuropathy. Again, this is a follow-up on that study, a new research together with a colleague at Imperial College London, Dr. Simone Giovanni, who had an interesting observation from a mouse model of peripheral neuropathy, where he noted that mice with the hyperlipidemia tend to have more severe peripheral neuropathy, especially the one that affecting the small nerve fibers that causes pain. And so we're basically looking to see if we see the same type of an effect in our patient cohort, both in the skin biopsies as well as also in their blood samples. And this is basically a very beginning study. As I mentioned, we've done the genetic study in the first 800 patients with our collaborators at WashU. But one of the other things is, the gene abnormalities is one thing, but they all have to be converted to proteins in your blood for those abnormal genes to do what they're supposed to do. And so one of the things we're trying to figure out is, are some of the gene abnormalities we see in these patient population, do they really actually alter the kind of the proteomic makeup of the patient, of their blood samples, and whether we can learn something about how idiopathic neuropathy causes nerve damage. Again, this is, I think we mentioned some of this earlier. We're doing a more detailed genetic study with the diabetes and CIPN. I will kind of pass on this slide. This is basically kind of an expected result. I always tell my patients, if you're tall, you're more likely to have neuropathy, and it's going to be more severe. The longer nerves you have, you're more likely to have damage. Also, as you get older, the incidence of peripheral neuropathy increases, and this was a study done together with Kansas University, and the students are trying to write up the paper. And so one of the other questions, you saw that earlier, some of our work was looking at vitamin levels to see if they are related to neuropathy. We looked at the diabetes and metabolic syndrome. But one of the other potential contributors to neuropathy is a systemic disease called hypothyroidism. If you have severe untreated hypothyroidism, we know that it can cause peripheral neuropathy. But if you treat the hypothyroidism, does it still cause neuropathy or causes it to progress more rapidly? And the answer is no. So if you have good, well-controlled hypothyroidism, you don't need to worry. And then another kind of a simple descriptive project that is ongoing is looking at the incidence of carpal tunnel syndrome or pinched nerve at the elbow, and whether diabetes increases the risk of that. And basically the paper is submitted and it's under review. I think I have only a couple more slides before I pass on to Adam. As I mentioned, one of our biggest interests is to get pharmaceutical companies more interested in this resource and help them develop treatments for patients. So one of those companies, Vertex Pharmaceuticals, is interested in looking at our diabetic peripheral neuropathy patients, especially to see how their diabetes is changing their neuropathy over time. So we have some patients where we have longitudinal data where they're seen a couple years apart. Another company, Veritas, is interested in, again, in diabetic neuropathy patients to see how their medication use changes. Scripps Institute looked at some of our samples, looking at something called Wallerian degeneration pathway. This is an important mechanism of how nerves degenerate in peripheral neuropathies. And in fact, multiple drug companies are now trying to develop inhibitors of this pathway. And then finally, one of the companies called Sangamo, who is developing gene therapy for painful idiopathic small fiber neuropathy. They were looking at our samples to see about the safety of their gene delivery tool, which is this adeno-associated virus. I think the future is really expanding this data set as large as possible. But more importantly, we need to start creating what we call induced pluripotent stem cell lines from patients with peripheral neuropathy, so we can actually study disease mechanisms directly in patient samples rather than trying to model the neuropathies in mice alone. And we can then, once we have large data sets where instead of looking at proteomics in 30 or 60 patients, doing it in all 2,500 or 5,000 patients, then we can start using machine learning or AI to really help us understand disease mechanisms and identify new therapeutic targets. And one of the goals is really making this research resources publicly available. That's important. I always talk about how the ALS field, Lou Gehrig's disease field, made this an amazing resource, and they have now over 10 different clinical trials ongoing, and they already have a couple of drugs that are approved, and they're not standing on their laurels, but trying to expand this genetic study that they've done in 5,000 patients, and they already have over 1,000 stem cell lines to over 10,000 patients and stem cells, so that multiple researchers can study this and use, again, the machine learning and AI to better understand the different subtypes of ALS. So with that, I think that was my last slide. I will stop and pass it on to Adam.
Thank you so much, Dr. Höke, for your coverage of the research that's going on through the PNRR and kind of our hopes for the future with that research. Yes, I'm excited to bring on, we have kind of connecting to that, Adam Halper, who is, as I mentioned in his bio, a member of our board, but also he is a PN patient and an enrollee of the PNRR. So Adam, I will send it over to you to chat about this from a patient's perspective.
Okay, thanks, Amanda. Thank you all for joining this, and it is a pleasure to speak very briefly. I think Dr. Höke should be the star of the show here, but very happy to share my perspective. Before I sort of dive in, and I'll try to hit the 4 bullet points that Amanda has listed on this slide, I would just say at the top that as a small foundation, we are very lucky to have some of the leading scientific experts globally on this condition working with us. And I think Dr. Höke is a prime example of that. We have a couple of other world-class leaders on our board and on our scientific advisory board as well. And so in my experience as a board member, it's been enormously gratifying to get to see their work in action and to get to collaborate with them. So a bit about my personal journey. I was diagnosed with PN in 2014, age 29, so relatively young for a PN patient. Had an acute onset. My neuropathy would be characterized as idiopathic, and I've been able to see a number of different neurologists around the country, including Dr. Höke. So I think we feel pretty confident in that diagnosis. Over the last 11 years, I'd say I've been somewhat fortunate that I've been able to manage the neuropathy fairly well, and feel quite grateful that I'm able to continue to live a life that is, I think, quite full in spite of the neuropathy. With that said, I felt about 9 years ago in 2016 that with my business training, I could play a constructive role in helping the foundation grow. And so I joined the board in 2016. I think of the Foundation for Peripheral Neuropathy as in some ways like a startup in the foundation world. And so what was interesting to me about getting involved was the fact that this was an organization where I really felt I could actually make an impact. And I think it's been somewhat slow and steady growth, but I think over time it's been exciting to see the foundation continue to grow, and the organization continue to increase its impact across a variety of levers. The other hat that I'm currently wearing with FPN now is as of the last couple of months, I'm our new development chair. And so I am now very much focused on thinking about how we continue to grow revenue for the foundation, which will in turn enable us to fund more research. In terms of why the biobank is important, so as someone who joined the biobank after an appointment with Dr. Höke, I think it's important because, as sort of Dr. Höke laid out in some detail, this is an infrastructure for us to fund research that can ultimately lead to better treatments. And so I think my view tends to be an efficient investment can often be an investment in a platform to do a lot of other good things. And so for me, it was important to join. In terms of the biobank enrollment, Dr. Höke, I think, laid out the steps. It was, at least for me, fairly lightweight. I think I was in their clinic for, half a day at most. So very lightweight. I think, I personally did not have any concerns around going through the process and ensuring that I was a PN patient that could contribute towards research in a small way. And in terms of the future of PN research, I actually think Dr. Höke probably covered that a lot better than I will. I will say, I think from the foundation standpoint, I am excited that over the last few years we've been building out an advocacy practice and lobbying members of Congress for more funding. We've had actual real success in doing that, and there will be a lot more to come there. And so I think part of what I'm excited about is just the growth of the foundation and our ability to advocate for more research funding. And then I'd say another thing that I'm excited about that Dr. Höke also hit is that we're living in a time of pretty unprecedented technological innovation. And so I think a lot of that will bleed through to biotech innovation over time as well. And so I think, I'm hopeful that the next decade or two, but we'll see a lot more innovation around PN treatments than the last decade. So with that, I'll pause. That's kind of my quick spiel. And Amanda, I'm happy to take any questions from folks that have them.
Great. Thank you so much, Adam, for providing that perspective, and welcome back, Dr. Höke. We've got some time for some questions, so I'm going to pull as many as I can from the question box from you guys. And some of these will be directed at you, Dr. Höke, and others at Adam. A couple, I think we could throw them to both of you. So feel free to interject whatever points you want, even if it is a question for you, Adam, and you want to say something, Dr. Höke, or vice versa. So I think the first question that I will pull from our Q&A is more focused, on some logistics of the PNRR and our biobank, and how does the PNRR and the PIs involved in the PNRR ensure the privacy and security of patient data within this biobank that we have?
Yeah, so all the biobank at each university, you go through an ethics review. So it was called an institutional review board, IRB approval. And part of that requirement is that we do not keep any identifying information about the patients. The patients basically are assigned a number, and all the data that is and the blood samples assigned to those patients are kept with that number alone, with no linking back to the patients. So I always get that question asked sometimes, the patient says, okay, I'm giving my blood, you do genetic study, and if you find something, will you tell me? And unfortunately, my answer is, I'm sorry, but I can't tell you back what your mutation is. The only thing is when we find a new cause of neuropathy and commercial companies now start using them, these new genes and new ways of analyzing DNA samples, then we can ask the patients to get retested. But we won't be able to, and that's one of the main reasons how we protect their privacy.
Great, thank you, Dr. Höke. Yes, and even the information we receive here at FPN is completely anonymous as well. We do not receive any information about the patients. Next question probably for you, Dr. Höke, as well. Adam, obviously, as always, feel free to jump in. So, Dr. Höke, how does the PNRR compare to other biobanks in kind of this rare disease field? Are there features that make the PNRR unique and useful? How does it compare?
Yeah, I think, there are, at least I would say in the peripheral neuropathy field, there are a couple other biobank attempts, especially in inherited neuropathy focused research groups. One of the limitations we saw there was patients were submitting their own bloods with their own diagnosis without careful characterization of the type of symptoms they had, or their exam findings, or even their nerve conduction studies were not done in a standardized manner. So when the researchers were analyzing those data sets, they realized that it was difficult to make sense of some of the data because if you don't have standardized, well-curated data set, it becomes very difficult to do statistical analysis and you have confidence in your data. And so that was one of the important things from the beginning. We wanted to make sure that we weren't going to kind of enroll patients from anybody. We wanted our data sets to be really high quality so that with a fewer number of patients, we could have enough statistical power to do the type of genetic and biomarker studies that we're doing.
Great, thank you, Dr. Halper. Adam, this next question comes from the Q&A for you. What do you think researchers often overlook about these day-to-day experiences of neuropathy patients?
What do researchers overlook? I think I don't know that I can answer that question that well since I'm not a researcher.
That's okay.
You know, I think what I would say is, you know, I think a best practice, and I imagine some researchers do this, is to try to take a sort of, you know, human-centered approach. So looking at the way that peripheral neuropathy actually impacts one's quality of life, and using that as an important input to think about what an optimal treatment is. I think good researchers do that. But I also think there's a lot of the work that Dr. Höke is discussing is sort of foundational work that one needs to do to even get to that second step. That would be kind of my quick reaction.
Great, thank you, Adam. And I think Dr. Höke, go ahead. I'll ping this question to you as a physician, as a researcher. What do you think researchers often overlook about these day-to-day experiences of neuropathy patients?
Yeah, I mean, I think I will answer that first as a clinician. As Adam mentioned, it's really important to really think about the patient, their quality of life and their overall experience in their day-to-day activities, and not just looking at the nerve conduction study results or EMG reports or lab results. I always tell students and residents, you know, you're not treating the test results. You're treating a patient, you're treating a human being. So you really need to focus on their symptoms and how they're responding to the treatments that you are giving. You know, currently we don't have any treatment that can reverse neuropathy, but for most patients, we can make them comfortable. You know, for painful symptoms, we have treatments. When patients have balance issues, you know, referring them to physical therapy or balance training exercises in a timely manner can have a huge impact on their quality of life. Coming to the question, you know, research phase, as Adam said, right now what we're doing is really very foundational. I think once we have drugs that can hopefully impact the trajectory of their neuropathy, that kind of prevents the further nerve degeneration or promotes nerve regeneration, designing clinical trials such that the end result is what the patient feels and experiences in their daily activities and not just correcting one simple thing in their measurements. You know, if I have a patient whose nerve conductions are improving, but their neuropathy as they experience is not changing, I'm not sure if my drug is actually good.
Oh my gosh. Ooh, I'm back. Sorry, I think my internet cut out. Did I cut out? I apologize, guys.
No, you're okay.
You're still here.
Oh, I'm back. Oh, good. Excellent. Okay. I never know sometimes if that happens. It said my internet went red and I didn't have connection. So great. Thank you, Dr. Höke, for kind of answering that question as well. My next question comes from the audience too. So kind of keeping in mind that the PNRR is not accepting patient enrollees with CIPN, chemo-induced peripheral neuropathy. Dr. Höke, can you recommend any other ways that these patients can participate in any research efforts?
I'm thinking if there's any groups that are enrolling patients. So there are some sites that are part of these Peripheral Nerve Society, which is this academic organization for peripheral nerve researchers. And within that, there's a Toxic Neuropathy Consortium. And within that, there are efforts to better study CIPN. So I think easiest thing to do is, if you're interested in CIPN research, is to log into the Peripheral Nerve Society's website and reach out to the Toxic Neuropathy Consortium organizers.
Wonderful. Thank you, Dr. Höke. Great. This next question is for you, Adam, but again, Dr. Höke, feel free to jump in. Let's do this question. Let's do this question that could be for both of you. What is one thing that you wish more patients understood about how their participation in the PNRR would accelerate treatments or discoveries of cures?
Adam, do you want to go first?
Sure. I mean, I think
if you guys need to take a beat and think, I can put another little plug into something I was going to say before my internet cut out. So go ahead, take a beat. I was going to say the fact that, you know, Adam and Dr. Höke were discussing how it's important to see patients as patients and not just as a small little fix for some sort of research aspect. And I think that that's something FPN we do really well with, focusing on our buckets of programming with our patient education and research and advocacy. As we focus on this research and doing webinars like this is based on this research, but also we have patient education and we focus on showing the lives of those patients. We do a lot of patient features, a lot of interviews with them and stories to really feature their lives. So I think that that is something that we do really well, and I really enjoy personally as program manager. So that was something that I wanted to plug in there. But okay, Adam, I'll send it back to you.
Sure. So I think there are a couple things that come to mind. You know, I'd say one is, like, when thinking about innovation in the sort of broader pharmaceutical space, one point to bear in mind is that timelines are never as quick as one might like them to be. So I think just have like coming at it with some degree of humility and understanding that there are a lot of really smart people looking at this problem, but progress is not going to be as efficient as one likes. So I think that's number one from a patient's perspective. But then I'd say second, and I think Dr. Höke walked us through a lot of that today, I think what's exciting is we've actually been able to use this registry to advance research to this point, and there's a lot more to come. And so I think to maybe try to leave with a little bit of a note of optimism, as I referenced before, I mean, this is a really exciting time from the standpoint of innovation, from a tech perspective, and a lot of that is going to bleed through over time to drug development. And so I am more optimistic today than I might have been 5 or 10 years ago. Curious to hear Dr. Höke's answer.
I echo your sentiments. I mean, I think at some of the other webinar setups, I mentioned this new drug candidate, the SARM1 inhibitors. The preclinical data in inhibiting this one enzyme called SARM1 looks amazing. It prevents chemotherapy-induced neuropathy and diabetic neuropathy completely in animal models. And right now, there are about 8 or 10 pharmaceutical companies, including big pharma like Lilly, AstraZeneca, Roche, are racing to develop SARM1 inhibitors. And 2 of them are already in phase 2 clinical trials, and which means amazing in such a short time. And if they are successful, we may have our first true disease-modifying therapy, like something that inhibits or prevents nerve degeneration going forward. It doesn't regenerate nerves yet, those type of drugs, but it will kind of stop the neuropathy from progressing. And that's why I'm really excited for our field compared to, as you said, 5-10 years ago. You know, we and others did some of the early preclinical work in this molecule, and it's really nice to see those coming to fruition. I hope we'll have a drug before I retire.
Great. Yeah, Dr. Höke, I think I can tell it's contagious, your excitement about this. Me not being a clinician, it makes me excited about this research that we have. And also you, Adam, I hope that this research too, and the future with the PNRR has given you some sort of hope and empowerment as managing your neuropathy and all of that. I also think that it's important that you touched on the timelines and making sure that you're patient with these. And I think that that's a lot of things that, myself included, we do not understand that, you've got different phases. Dr. Höke, you mentioned different phases in these studies, these clinical studies. And I think that it takes time. But these are really exciting things that we have going on. And then, so we're about at time. Thank you all for all of your questions. The last thing I just want to put on both of you, is there any other last, takeaways? What's one takeaway you would love to hear or give our audience that we have, or any hope that you see for the future specifically of PNRR, where you would like to see it go?
Yeah, I mean, I would say if you're a patient with peripheral neuropathy, reach out to centers, reach out to the Foundation for Peripheral Neuropathy, find out which centers are participating in this, and be a participant. Donate an extra tube of blood and fill out a questionnaire along with getting your detailed clinical examination. One thing, you'll get probably better care and also participate in this important research. So I want to thank all the participants who really helping us, put the foundation for future research.
Thank you.
I don't know that I have anything else to really add there, but maybe just a quick plug for the foundation is, we're always looking to grow, and I think if you're interested in getting involved, I'd reach out. I'll save my development plug for another time, but we're small and growing, and I think there's going to be a lot of exciting work ahead.
Yeah, definitely. Yeah, small but mighty, right, Adam? Small but mighty. Right. I agree from that standpoint. Yes, and if you, to echo what Adam said and Dr. Höke, if you are interested in learning more about the PNRR that you learned today, we do have more information on our website, or you can reach out to us at the foundation for more information about getting started and looking at our website. So thank you to everyone who has submitted questions. Our viewers, we had a lot of them, so we got through as many as we could. And then obviously Dr. Höke and Adam, thank you again so much for taking time out of your busy schedules to be here with us. To our viewers, again, if you want updates on our upcoming webinars like this, sign up for our newsletter. You will have kind of first view of any of the upcoming webinars we have, and you can register right away. And also, if you want to ensure that we continue doing webinars like this, donate today. Anything you can contribute helps, continues with research. It helps with research, advocacy for PN, and also patient education, which are all things that we at FPN are working towards. So thank you guys so much again. I appreciate it. And we will see you all again soon. Thanks, Adam and Dr. Höke.
Thank you.
Thank you.
Bye.